All right, good morning slash afternoon slash intermediate time. It's nice to meet you, Melissa. Do you want to say hi and introduce yourself?
Yeah, hi. I'm Melissa Cooper. I'm a postdoc at NYU Medical Center over in Manhattan, so almost all the way across the world from where you're sitting right now.
Awesome. Well, lovely to meet you virtually over Zoom. And so the reason we're meeting up today is to discuss a recent paper from you and the group you work with, which was really beautiful study of astrocytes, this really fascinating cell, specialist cell in the nervous system, and in particular, the really interesting connections between astrocyte cells that kind of allow signals to sort of process over large scales. So, do you want to maybe give us a little summary of kind of what your study was about and the kinds of things you found?
Yeah, I think astrocytes are still kind of the Wild West in neuroscience. There's still like so much we don't know about them. And I think for the longest time, everyone's just envisioned them as this like housekeeper cell that takes care of the neurons right next to it and then lets them survive any kind of damage, but then kind of doesn't really do much day to day. But an astrocyte really is this really important cell that has a lot of connections across the central nervous system through these channels called gap junctions. So, gap junctions are these little pores between cells. And I think if you go through the literature, you'll find like these really conflicting points. You'll see some people that think there's this big astrocyte syncytium where every astrocyte is connected to every other astrocyte so that you can just like diffuse things out through this vast pool. And then you have other people that are looking at astrocytes as these like very local networks where like waves of calcium might go from like an astrocyte to a neighboring astrocyte. But like all of that specificity that people have been thinking about has been local. I've done a lot of work in neurodegeneration, and I kept finding that astrocytes in specific regions on the other side of the brain or really far away from a place when I had like a very local injury would start becoming a little bit different, would start responding to that
Mm.
injury, but not everywhere. And so I started to think about how they might be interconnected or share resources. So I really wanted to get into how networks of astrocytes are structured across the brain.
Awesome. Well, maybe we'll get to the neurodegenerative question at the end. I have a bit of a
Mm.
background working in Parkinson's disease, so I'd love to pick your brain about that. But perhaps before we get to some of that nitty-gritty, maybe we could zoom out a little bit. And for those in listening or reading that haven't really come across this term gap junction before or know
Mhm.
what makes it different from traditional synapse, can you maybe say a little bit something for us here about what a synapse is and how a gap junction differs?
Yeah, you'll see in the lit, sometimes people refer to a gap junction as an electrical synapse.
Hmm.
So chemical synapses are the ones we normally think of between neurons, where one neuron releases chemicals onto the next, and that causes that other neuron to behave in a different way.
Hmm.
These gap junctions are just pores. So each different type of gap junction, you'll see like connexin 36, connexin 43. That just is the molecular weight of the protein that makes up the gap junction, and it makes a different size pore. And it just so happens that astrocytes use the gap junction that has the biggest pore of all of the options that were available.
Awesome. Cool. And so these pores are fascinating because in some sense you don't need to have like a little packet of neurotransmitter that's sitting in a vesicle that a signal comes
Mm.
down and then you release it across a junction and then it hits a receptor. Now what you have is a direct connection between cell A and cell B, and that could be their cell bodies or it could be their dendrites or maybe the axon initial segment, or I don't know what the different terms are. I guess it would be the little parent, I don't, what are they called? What are the little branches of an astrocyte
You
called?
just go
A
processes. Like
process. Okay,
that's,
great.
yeah.
So it could be sitting on their process. And the idea is that whatever happens in one part of that one cell is now kind of contiguous with another cell. And so you can get this kind of like almost passive transmission of a membrane potential change or a particular ion concentration or something like that. Is that the idea?
That's the idea. But then I think you also might be tricked into thinking it's only ions that go through. There's
Mm.
some really amazing work done, especially in cancer, and you'll see some in like cardiac work as well, that show the things that go through gap junctions actually, especially connection 43, can be up to like 2 kilodaltons in size.
Okay. Can you give
So
us
like,
like a vibe of how big 2 kilodaltons is for those of us who don't think
yeah.
on the nanoscale?
So as we're thinking about calcium, if we're thinking about calcium moving through or different ions like potassium, stuff like that, you're thinking
Mm,
about like tens of daltons. If you're thinking about glucose, lactate, pyruvate, you're thinking like around 100 daltons. As you're starting to think about peptides, like each peptide you can think of as maybe like 150 to 200 daltons in size to give you
wow.
an idea of the scale of what can actually move through.
Okay, so this is like an airplane hangar door that we can drive things through.
It's
Cool.
bigger than I think a lot of us realized it could be.
Yeah. Okay.
Yeah, yeah.
Very
It's
cool.
gated, so like they can control how big that pore is to kind of like
Yeah.
constrict what goes through. And it's still a huge mystery what it is
Yeah.
that's going through. But the machinery that I'm using to do this marking of what's going through gap junctions actually relies on an amine group, which is going to be
Oh.
on some of these like bigger molecules going through your gap
Ah,
junction.
this
Hmm.
this this warms my biochemical heart. I was my
Hmm.
undergraduate was studying biochemistry, and I loved how when you dove down into the details of these little kind of mechanisms, right? Like there's a an amine group that's sitting there, and it's got free electrons or something like that, or it's actually no, it's the opposite, right? It's got a little positive charge on it,
Yeah.
and that means that anything with electrons wants to come and get close to it, and it hates anything with a positive charge. I love that these kinds of details down at that level actually have like a physical
Hmm.
relationship with the kinds of milieu around them. And oftentimes, when we're sort of studying things up at the kind of more micro or even meso macro scale, we kind of forget about this real physical kind of logic that's down at that scale. And
Yeah.
it's really fun that you get to connect with that. And the kind of imaging you do, you can see it, but you actually know that
Mhm.
it's related to those things. That's really neat.
Yeah, but I think one of the most fun things about this paper is if you look at the scale bars, I have scale bars in angstroms and scale bars in centimeters. And that just
Awesome.
brings me so much joy to like be able to see the brain at all of these different levels.
I love it. That's fantastic. Okay, cool. So we have astrocytes. They have these really interesting gap junction airplane hangar connections between one another. It's not exactly a, all to all, everything's connected to everything. It's also not just the fact that they're, connected or disconnected in some local circuit. It's somewhere in between those two.
Yeah.
And you were interested in trying to characterize that, understand precisely what the project. Tell me more about what you were interested in at the start of this project.
Yeah, I think there's a lot of mysteries that are still open in neuroscience. I think one of the really cool ones you'll see is that in stroke and in a lot of different diseases, when you have like on one side of the brain this perturbation that happens, astrocytes in the same region on the other side of the brain start responding.
Mm,
And like this has been a huge mystery for a long time, but it actually is quite a big deal. It matters to at the level of like us designing experiments. A lot of the time you're using the other side of the brain as an internal control.
huh.
And it's actually not naive. These
Mm,
things are all
mm.
interconnected, just not in the way we thought. They're connected by astrocytes.
Yeah,
But it also
fascinating.
matters, like, because there are certain brain regions that consume so much energy all the time that it would
Mm,
be physically impossible for the astrocytes right there to be able to keep those neurons just like functional all the time. They have to draw upon energy from something else. So I
mm.
started thinking about like energy transfer between regions and the specificity that might be happening because you might want one less active region attached to one more active region so that those astrocytes can support it. All of these things that the brain might be doing.
Yeah, that's awesome. So tell me a bit about how you went about studying this. You had an access to, I guess, a connexin
Mhm.
genetic line, and then you were able to kind of manipulate that to image the cells. Can you tell us a little bit about that process?
Yeah, I think the exciting one that I love is that I built this vector, and it's kind of this idea, marrying two ideas a little bit.
Mm,
So most of the way that people have studied gap junctions up until now is you'll take like a slice of the brain, touch an astrocyte, and diffuse biotin
mhm,
across. And biotin's like 250-ish daltons, so you can start seeing it move across.
Hmm.
all of these gap junctions. But now you're looking in 2D because you've only got one slice of the brain. And when you're slicing it, that's an injury, and astrocytes respond to injuries. So this
Hmm.
isn't perfect. And then there's all of these tools now where you can get really fast biotinylation of things that are right next to an enzyme. So I was thinking about all of these tools where we're biotinylating things that are right near that enzyme. And then the fact that biotin has been what people have been using to mark
Hmm.
what is connected through a gap junction functionally
Hmm.
for a long time. And I just attached that enzyme to the gap junction. So now
Awesome.
whenever something gets there, it gets biotinylated. And now I can track it as it's moving in the awake, behaving, alive mouse and see where it
Awesome.
ends up.
So, like, you put a little sensor on our airplane hangar so that anytime anything
Yeah.
drives through, it lights up a
It's like
little
it's a
light.
stamp. Yeah,
Yeah, a little
it
stamp.
gets like a
Okay.
stamp on it. Yeah.
But then another part of your study that I really loved is that you didn't stop there. You could, I guess, you could have done that with an animal, and you could have waited a little while, and then I guess you could have, killed the animal and then sliced their brain up, and then look to see in this slice or in this
Hmm.
slice or in this slice do I see biotin? But you didn't. You didn't do that. You did something kind of different. Do you want to tell us a
Yeah.
bit about what that was?
Yeah. So I optimized a clearing protocol that's a lot like iDisco, but I wanted to optimize it to really maintain astrocyte morphology. So I just
Okay.
made it a little bit more gentle, but
Okay.
uses the same principles. So I now clear the brain so that I can image the whole thing, each cell in an entire mouse brain, and see all of the biotin that's moved across different astrocytes inside of
Awesome. And for those that are interested, you should check out the supplementary videos in Melissa's paper
it.
because they're just fantastic. This
Yeah.
beautiful kind of sort of black background, and then all of a sudden this bright pink kind of pops up, and then you see these nice, beautiful patterns spreading
Mhm.
around. And as someone who doesn't spend a huge amount of time looking down microscopes like you would, Melissa, it's really striking
Mhm.
to see these kind of beautiful distributed networks that pop up when you go and have a look. So please describe to us what was it like when you first looked down the microscope and saw
Oh my God.
these images?
Yeah, that moment. Oh my goodness. It was super late at night. I was alone on the microscope. I had like cleared
Nice.
the brain earlier that day, and normally you like wait overnight, and I just couldn't wait. I like went back.
Nice.
I was just so excited. And I remember putting the brain on the scope and taking a look and seeing like I don't know what I was expecting. I was thinking I might be getting like, this ball of astrocytes everywhere, like the syncytium we were talking about, or just
Mm,
start seeing these like more local networks. But I was thinking about some regions right next to each other, maybe talking. And then I saw these really specific chains of astrocytes that were like holding hands going across corpus callosum all the way to the other side of the brain. I just remember like shaking. I had to like adjust the brain and like my hands were shaking. I lost an olfactory bulb on the front of the brain. So like now whenever I show, I actually like in my talks, I show this like first brain that I ever imaged. And if you look really
Oh,
closely, you'll see like one olfactory bulb isn't there.
that's awesome. Oh, that makes me
Mhm.
so happy. Just as a meta point, I think I love to say to my students how important it is to find the wonder in what you do. And it seems
Mhm.
like you found the wonder that night,
Oh, it was so
late,
cool.
dark in the microscope lab. So that's really awesome. Um,
Hmm.
cool. And so you have these beautiful networks, and then I think another really striking thing as someone from outside the field is that if you seed areas of, say, motor cortex or somewhere like the prefrontal cortex, so I think even the hypothalamus, you see
Hmm.
these very different, distinct networks. They're not exactly non-overlapping. Some parts are
Yeah.
common amongst both, but other parts are different. Do you mind saying something to us about that? How you did you start with one of those areas and then pick another, or how did you go about choosing
Yeah.
those regions?
Yeah, so I picked like a few different cortical regions when we were starting, and I just noticed motor cortex seemed to be really interconnected. And
Okay.
it was a nice place for me to hit too, right? It's like very central as far as cortex goes. So then
Hmm.
I can like very easily keep replicating that same injection site. But I think it's
Gotcha.
also just a really useful spot for people to start looking.
Okay.
In the paper, you'll see a lot of the images are from either motor cortex, prefrontal cortex, or hypothalamus.
Hmm.
And so I started in motor cortex, just picking this like very central cortical region. And then I wanted to see if another cortical network would look the same. So I picked a cortical region that was far away and had a really different function, so prefrontal cortex.
Hmm.
And then I was thinking, I'm just sitting here in telencephalon. Now let's move into diencephalon and see if the two are going to talk to one another and if those astrocytes have similar networks. And it
Mm.
was really funny because the motor cortex network is very different from the prefrontal cortex network. The
Mm.
prefrontal cortex network is actually overlapping quite a lot with the hypothalamic network,
Mm.
which is not what I would expect. It's almost as though we like hit two sides of the same highway, perhaps.
Yeah,
Uh,
nice. And I guess maybe this is another area where I was just uncertain about how all this worked. So when you injected the your little biocytin tagging mixture, you had to wait an amount of time. And this kind of
yeah,
reminded me of some of the methods that they used to use back in the day with like attenuated viruses, and they would inject the virus, and then they might do a transsynaptic jump or something, and they'd wait, 4 days for one synapse and then 10 days for two synapses or something like that. Is it
yeah.
similar to this where you've got to you've figured out a strategy where if you wait a certain amount of time you can guarantee that it will have moved, but if you wait too long maybe it all kind of spreads out everywhere? Like how do you figure that logic out?
Yeah, so the virus stays in one spot with the one I'm using. I'm
Okay.
just infecting like one population of astrocytes and making it so their gap junctions just tag everything as it goes through. Um, Most of the waiting process I did was I wanted to make sure the mice were totally recovered from the surgery because
Ah,
astrocytes are going to be one of the
because they're
things that's going
reactive.
to exactly. So I wanted to make sure I was looking at a homeostatic network, at least as I'm mapping out these initial ones.
Gotcha.
So, like the proteins in an AV, you'll probably get like pretty good expression a week or two after you
Mm,
do the infection, but I was just being extra careful. So I
awesome.
gave it 3 weeks and then I wanted to see like everything they would be connected to. So the other cool thing about this is you get temporal control. So you give biotin in the drinking water, and that's when the biotinylation starts.
Mm.
You can decide when you want to start seeding your network. You could decide to do it after a mouse learns something or after there's an
Ah.
injury, and then give biotin and see that kind of network specifically after that
Awesome. And that is a fantastic segue to another really awesome, fun thing you found in your paper, which is that you manipulated the mice's whiskers. I think it's you sort of trim the
happens.
whiskers, and I guess then they become less interesting sources of information for the mouse.
Yeah,
They're not
it's like
wiggling
a
around as
sensory
much,
deprivation.
a
Yeah.
sensory deprivation or something like this. And then you re-imaged the mice after they'd had their whiskers twigged for a certain amount of time. And then you
Hmm.
kind of compared the astrocyte network before and after the haircut. And you found big differences. Do you want to maybe tell us something about what you found?
Yeah. It was actually like two groups of mice, one
Ah,
that got the haircut and then one that didn't.
okay.
So then we can compare the two just because we have to kill them to be able to see the network itself. Um, but then I could at least compare between these two groups and see exactly what the networks look like. And the network is like half the size when I'm looking at it after the
Wow.
sensory deprivation. And we have so many ideas of why that might be. Like
Mhm.
maybe these neurons just need less energy, so you're going to have a smaller network. The way the network shrank was also really interesting because at least when you look at the way neurons remodel in this same paradigm,
Mm.
usually it's like their dendritic processes will all like shrink down,
Okay.
but their axons are still going to connect to the same place.
Okay.
The astrocytes started, they shrank down the network, but they were also connecting to different places. So
Huh.
the way that astrocytes remodel is also different than the way neurons remodel. So it's like we can't infer what an astrocyte network will look like by looking at a neuronal network, and we can't infer how an astrocyte network will remodel by looking at how neurons remodel.
Yeah, that's super cool. And can you tell from the kind of level of microscopy that you have access to here, whether it's the parts of the astrocyte that are connected to synapses that kind of wrap around a synapse, or like the
Mm,
pre and post synapse, versus the ones that connect to the end feet of the smooth muscle cells of the vascular system, or is it kind of
Yeah, I
hard?
can't tell at this level right now. It's something I could go back in. Like the cool thing about this clearing method is that you can actually like rehydrate the brain and section it and then go image it like later.
Oh, cool.
Um, so you get to like I have this whole library of these now. So then if I had like have a question later, I can like pull the same sample, store it at room temp, pull that same sample, have the image of the entire brain, rehydrate it, stain for something else, and then go
Wow.
take a look.
Oh, that's
So
awesome.
yeah, and it's great too because it just makes each sample, you can get so much more information out of it, which just to me, I really love that because that means we just need to use fewer mice now. We can like actually extract a lot more out of each sample. And
Yeah.
it makes it a bit more accessible too, because it's just less expensive when you have to use fewer things to be able to answer more questions.
Yeah, that's fantastic. And so
Mhm.
one of the other things that I found really fascinating about the astrocyte field is this really cool sort of emerging understanding of the complexity of the neuromodulatory system's interaction with the astrocytes and the astrocytes in the context of other neurons. So there was, I'm sure you saw this, but there was a beautiful paper the other day showing that the astrocytes themselves actually swell up and dehydrate as a function of arousal state. So if you're in kind of awake,
Mhm.
there's a big fat swollen astrocyte kind of sitting around, and then when you go into kind of non-REM sleep, it sort of diminishes down and becomes relatively desiccated.
Mhm.
And this idea kind of start dovetailing with this really beautiful work like Mike and Nedergaard's work, thinking about
Mhm.
this sort of lymphatic system swelling during stages of sleep. And I just think there's such a really beautiful little triangulation of different fields, all sort of feels like they're
Mhm.
coming together in the same space. So
Yeah.
I'd love to hear if you have thoughts about how neuromodulators might affect the spread of this gap junction connected network, or if you think that there's cool interactions there to understand better, or how you go about understanding.
Yeah. Oh my gosh, there's so many cool things here. So I think in the paper, one thing I want to point out is I actually perfused the mice at the same time of day, every single sample that I took,
Nice, nice.
because I think, especially from Miken's work, we can see how different the cells are going to be at different times of day. And
Mm,
so I bet this is a network that's going to change if we're looking day versus night. And I think perhaps, I don't know, under anesthesia, there's all of these really interesting
yeah.
different ways of modulating the central nervous system and its arousal state broadly. And I'm sure that the networks might change as we're looking in at all of these different things. As far as neuromodulation, there's all of these amazing papers that have come out recently that are starting to like incorporate astrocytes into neuronal circuit diagrams.
Yeah.
And I think the next step of that is going to be thinking about astrocyte networks and how they might perhaps co-regulate different neuronal circuits.
Yeah, awesome.
Yeah, I think there's so much open to explore in that way.
Yeah, that's excellent. I just was reading a really beautiful review paper by Andrea Volterra's group, and they have this lovely figure towards the end that kind of like highlights the multi-scale kind of system nature of astrocytes. And they've got the kind of the discussions about nanoscale. There's like a particular astrocyte and has connections to this and this end foot and this synapse or something, and then you can kind of like zoom
Hmm.
out to sort of microscopic level. Now you've got an individual astrocyte maybe connecting to a local network. Then you can kind of like zoom out even further and you get out to this kind of level that you've really beautifully imaged, which is now you've got these distributed networks of these astrocytes that are now coordinated across, I guess millimeters to centimeters in a
Hmm.
living brain. And I think I really love this idea of using these new imaging techniques and these kinds of inventive solutions to problems like you so nicely showed in your paper to kind of expose us to just how nuanced that complexity is. And
Yeah,
I really love this sort of new frontier for students that are excited. There's all these awesome opportunities to understand this,
yeah.
amazing system. I guess a mouse's brain is really small, like in your hands. It's still so complex and so difficult to
Yeah,
understand. So I really
there's so
admire what you're
much
doing.
there. Oh, thank you. I think it's been so fun to work in Glia. I just feel like there's so much right now that's available for us to find, and neuroscience is just a field as a whole where there's just been so many unanswered questions for so long. And glia represent a huge portion of the brain, and we know so little about them. So it's almost like just pick something you're interested in and think about how glia might be there. And you're going to be like looking at something that no one's looked at before.
That's awesome. So, I love that blue sky picture, and but instead of stopping there, I actually think I want to take you one step further, which is that I actually think is something you brought up very early on in our talk today, was the idea that there might be hints in this system's complexity that might help us understand this sort of odd, strange situations that often happen in clinical scenarios. So you mentioned stroke before. There are other disorders like neurodegenerative disorders like Parkinson's disease that have these transsynaptic spread sort of explanations for the spread of things like tau or alpha-synuclein, or that's in the case of Alzheimer's, or alpha-synuclein in the case of Parkinson's. Is there, I guess there's must be research on this, and people must be thinking carefully about how astrocytes would play a role in that process, and how an
Yeah.
understanding of that would help us get a better, appreciation of what's actually going on in the pathological brain. Do you want to maybe say something about
Yeah. I think there's certainly a ton of work like on the cell biology level, like the single cell sequencing, the
that?
RNA to protein levels. We know a decent amount now about how astrocytes work. But I think the idea of astrocytes being connected on this broader level is still now going to be an open thing as we're
Awesome.
looking at these different diseases. Things like tau and alpha-synuclein, those things are too big to go through a gap junction.
Gotcha.
But I think there's still a lot that can go through, like all of these
Mm,
metabolic waste products, metabolites themselves. And so astrocytes are still having to use all of their normal systems as they're cleaning up all of these things that are going wrong in disease. And so we can start looking at how like these normal systems might be becoming dysfunctional and maybe that contributes to the acceleration of a disease. So just because they're too big doesn't mean that the astrocyte networks aren't going to be affected.
Awesome. Well, Melissa, that was absolutely fantastic. Thank you so much for taking the time to walk us through your beautiful work. Is there anything you'd like to leave anyone with as a final thought?
Yeah, I guess this like kind of was a project where I was thinking so much about like I think the field of neuroscience has finally gotten to a place where we appreciate that glia are important. I think that's become everybody agreed now. I think as 10 years ago, like I was at SFN last session, last day. Now you've got me like giving a talk in the like prime time, which is so exciting. But I think like us as glial biologists, we're still sitting in a place where a lot of what we do is less useful to where the field of neuroscience is going. The field of neuroscience has been moving in a systems direction for a long time. And the field of glial biology, just by lack of tools and lack of people, we've been like really digging into this cell biology side of things. But
Hmm.
we haven't caught up to a place where what we're looking at is useful for everybody across neuroscience. So this is me trying to bring astrocytes to a place where they're more useful for systems neuroscientists to be able to think about how these networks of astrocytes might relate to this neuronal connectivity that they've been thinking about for a long
Awesome. Thank you so much for taking the time. Really appreciate it.
time. Yeah, thank you.
The Transmitter.